Can somebody confirm you can hear me?
Yes.
Thank you. Sir, first let me apologize for being late. I had to re-log in again because I accidentally shut down the Canvas website. And it took me 4 or 5 minutes to log in. So that just tells you how long it is to log in. Because like I said, you need to verify yourself twice and uh, and all that. Are there any questions before we begin? All right, if there's no questions, let me first state my computer is running a little slow. I guess you can't see that. I'm 96% done with the lesson for the lecture. Let me open this so you can view the page. There we go. 98% done now. So very soon I'll have a faster computer. Come on. All right, what we're going to talk about today is Lab 01. The file just finished. Um, Ubiquity of microorganisms. Please understand that the worksheet for Lab 00 and Lab 01 is due Friday at 8 a.m. If you do not submit the worksheet, I will drop you from the class because if you don't submit the worksheet, I do not have any idea whether you're attending or not. And the college does want me to state who is attending and who is not. And then they will drop the person who is not attending. You also need to read the infectious disease research project called Project Instructions, which you can find on Canvas for next week. I'm going to talk about that next week, and you need to be prepared and read it in advance. Let me show it here. So what I'm supposed to do, come on, is just close that page down, but not close Canvas down. Let me scroll down a bit. I think it's week 2 lab. There it is. The Infectious Disease Research Project. So I'll read the instructions and then I'll talk about it next week. Any questions about any of that? Oh, it looks like you got to download that one twice. Yeah, this is a turn in page. That's why I've got 2 links there. All right. Um, if there's no questions about that, let's begin with lab 01, the ubiquity of microorganisms. There is some pre-reading to do before this lab. Not too much for this one. A few pages discussing culture media, and then a page talking about obtaining pure cultures. There is some supplemental material in this lab, meaning video clips. The learning objectives for this lab is 1, know that microorganisms are almost everywhere. 2, describe the purpose of culture media. 3, describe what colony abundance means and what colony morphology means. These are 2 terms that we will discuss in this lab. 4, be able to define the following terms: inoculation, ubiquity of microorganisms, what's something that's sterile, what is isolation, what is incubation, what is in vitro, like in vitro incubation. What is a culture media? We have different types. There's a broth, there is a solid auger, there's a slant and a deep, and I'll show you those in a minute. What's a fastidious organism? What's a non-selective media? What does ubiquitous mean? And what's a colony? Any questions about any of that? All right. We've already stated that microorganisms are organisms that are too small to be seen with the naked eye. These do include organisms, cells, viruses, and prions. Even though viruses and prions are not something that are alive and they do not have cells. So when I call it, I'll call it a microbe. We will also include helminths, worms, in our studies, even though the adults are not microscopic or generally are not microscopic. However, their infective stages are microscopic. Another reason why helminths are included in microorganisms is microbiologists study organisms which cause human disease, and the helminths, the worms, do cause human disease. Any question about any of that? Come on, mouse. I'm not able to get my mouse to work. There it is. The phrase ubiquity refers to the concept that microorganisms are almost everywhere. Something that's sterile is something that is free of living cells. In the lab, usually when we have something that's sterile, it's because it was put in the autoclave to sterilize it. In this lab, we're going to investigate the abundance and diversity of microorganisms in the common environments. We'll also learn some basic lab protocols. Any questions about any of that? All right. A culture medium, let me put this down here and see if that works better. A culture medium, also called a growth medium, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms in a lab. Culture media can either be liquid, these are called broths, or are commonly used, and the broths are commonly put in a test tube. And we can add agar to the liquid media to result in a solid media. The agar can be put in a petri dish, but it can also go in a test tube. We don't usually use the broths in a petri dish because they're just too easy to spill, but they could be put into a petri dish. There's a video you need to watch for preparing how to prepare a pour plate. And note how the culture medium needs to be sterilized prior to its use to pouring the plate. An important question to consider is why must a culture medium be sterile prior to it being used? Can anybody answer that? We wanted to grow up some microbes. Why do we need a sterile medium before we grow the microbes?
So the results can be accurate?
Yeah, if you grow your microbes in a media that's not sterile, there can be multiple things growing in the media, and your results may not be accurate because the things growing could give you the results and whatnot. So we use a sterile medium, and then we put usually one organism in the test tube or the petri dish. So here we actually have a liquid broth in a test tube. This is a solid broth, both of these, and a test tube as well. And these are solid broth put in a petri dish. We usually call these agar plates because the agar is usually what's used to make the media solid. And you can see in the test tube, if we have a solid media, it can go across like this. This is called a deep, where you just pour the agar in the tube, let it solidify, and then it goes straight across. Or you can hold the tube at an angle, in which case you can get a slant. Any question about that? All right, today we're only going to talk about one media, a general purpose media. A general purpose media is non-selective. It usually includes complex composition, meaning has many things in it, and they support the growth of most non-fastidious organisms. So a non-fastidious organism is something that can grow fairly easily. A fastidious organism is something that will not grow in a general purpose media. You need to add certain things to a fastidious organism in order for it to grow. One fastidious organism is Streptococcus pyogenes, the bacteria that causes strep throat. It will not grow in a general purpose media because it needs certain nutrients which are lacking in a general purpose media. And usually it's a organic growth factor that is lacking. Some examples of a general purpose media include, I'm going to start down here, number 3, nutrient agar or Na, tryptic soy agar or TSA, and then nutrient agar with yeast extract added, and that we call NAY, Naye. These are the 3 most commonly used media in microbiology. They grow a lot of organisms, any that are non-fastidious, and all of these general purpose media are non-selective. Any question about any of that? Like if you wanted to grow E. coli up, you could grow it in any of these 3 general purpose media. The same with Staphylococcus aureus. It'll grow in any of these 3. All right, before we grow something in the lab, or in this case in our online lab, the culture must be labeled. So it is vital that a Petri plate or a test tube is appropriately labeled prior to its use. The following 5 items must be included. You have to have your name or your initial. You have to have the date. You have to have the type of medium used, like you can put Na for nutrient agar with yeast extract, or Na for nutrient agar. You have to put the inoculum, and that's what you're trying to grow, what you're trying to culture. And that can be something like E. coli, or if you're simply exposing the media to the air to try and grow things from the air, you just say something like air exposure. And then you have to have you don't have to have the incubation temperature is optional. So at least 4 of these things must be included on every tube and every petri dish that you're growing. And that way, if it's ever found, we know who to give it to. We knew the date that they started it, what media it is, what's growing in it, and perhaps the temperature. Like I said, the temperature is optional. Like here's a plate here, got the instructor's name, the organism growing, the date, the media, and there we see even the temperature that it was grown at. Any question about any of that? Normally, when you're writing on a petri dish, you should write along the edge so that you're not blocking what you can see in the I don't know, the center or the large portion of the center of the plate. So you generally write it along the edge. On a test tube, you generally write on a piece of tape and then stick the tape to the test tube. Inoculation is the placement of the microbial specimen into the sterile culture medium, meaning that's when you transfer the microbial specimen into the tube or the plate. Incubation is the placement of the culture media in an appropriate physical environment for a period of time to allow the growth of the microbes. Normally, in the lab, we incubate our cultures in an incubator. An incubator is just where we place them to incubate. Normally, the incubator that we have can control only the temperature, the humidity, and the time you incubate it for. We have very simple incubators, but you could have a more complex incubator, which can control the amount of oxygen and the amount of carbon dioxide available to the microbes. I've got a picture of a complex one down here. So there's a complex incubator. You have the different dials for changing the CO2 and the oxygen the microorganisms are exposed to. You then shut the door and the incubator warms the culture, either the plate or I don't see any test tubes there, so we'll just say plate. Those are dishes, but that's the plate. We can control the humidity by adding water here, and then when the moisture of the or the heat of the incubator warms up, the moisture will evaporate from this, adding humidity to the culture. That's actually important because if you don't have water in the incubator, then the culture cannot be alive in the incubator for as long as when you have uh, a source of humidity. If you have humidity there, you can keep the culture usually alive for a week or longer in the incubator. But if you don't, the plate or the test tube will begin drying out after only a few days. And then the time is just when you put it in and take it out. Let me see if I have anything else to discuss before going on. All right. When you're putting plates in the incubator, you should realize that the incubator will warm up. I'm trying to get a plate here. So that if we had our auger down here, can you guys see this? Anyone? Can you
Yep.
see this?
Yes.
Okay. Uh, and then had the lid on, the water will come out of the auger, condense on the lid of the plate, and then drip down into the plate. That may not contaminate the plate. In fact, the plate should be sterile. The exception is if the water were to drip down I don't know if you can see that, the edge of the plate and then fall into the plate. That likely would contaminate the plate because the very bottom of the lid is not sterile. Oh, there, I can see it there. As long as I put it by my face, I can see it. But most of the water will be dripping from the lid into the plate, and that will only mess up the plate, spreading the bacteria. If you had isolated colonies, you'll then have a whole bunch of growth, and you won't have isolated colonies. So therefore, all plates, when you put them in the incubator, they should be put in the incubator upside down. Now, the auger is on the top, And if water were to come out of the auger, it will condense on the auger. The auger will just wick the water so it won't ever drip. And even if it were to drip, it's dripping on the lid. Who cares? We don't grow any organisms on the lid. So whenever you incubate plates, they should be put in the incubator upside down. And then the advantage is you normally write on the bottom of the plate. I need to talk about why you do that. And that will be something you can then easily read when you look in the incubator, when you're looking for your plates, for example. Any questions about any of that? All right. Let's talk about why you write on the bottom of the plate. And I've got a very good example. There was once a lady that I was working with in a lab, and she had a whole bunch of plates from an experiment, and she was moving them from one place to another. And along the way, she tripped, and then all the plates dropped on the floor, and then the lid separated from the bottom of the plate. Uh, the trouble was she had written on the lid what was on and in and what the plate had been treated with. And so the experiment had to be thrown away because we didn't know which bottom where the growth was went with which lid. And the experiment had to be thrown away and repeated. So what you should always do is write on the bottom of the plate, because that's where your growth will be. And then if the lid gets separated from the bottom of the plate, you have the information for what's in the plate. Any question about any of that? So you always write on the bottom of the plate. And then when you put the plate in the incubator, you always put it in upside down. And that's to prevent condensation from forming on the lid and then dripping down into the plate, which could ruin the plate. Like if you're trying to get isolated colonies, that would ruin the plate. Any question about that? All right, the visible bumps on the agar surface, each of those is a colony. Bacteria colonies tend to be rather smooth textured, and that differs from a fungal colony. Each colony can have tens to hundreds of millions of cells. In fact, to see a colony, the smallest colony you can see will have at least 1 million cells. And that could be, all of the cells in that colony could be descendants from one cell, although they could be descendants from two or from a clump of cells. So let's look at no, I got a little video here. What's that video on? Oh, binary fission. I'll talk about that in the lecture. Colonies are only visible on a solid medium. They are not visible in a broth culture. You cannot grow colonies in a broth culture because in a liquid culture, all the cells will mix together. You will not have individual colonies. Let's come down here and take a look at the difference between a bacterial colony and a fungal colony. Well, I've got this someplace. Maybe I don't. I thought I did. Oh, here it is. I went past it. All right. Let me blow that up a little bit. A bacteria colony tends to have an even edge with a clear, distinct boundary between the colony and the growth. A fungal colony has a non-distinct edge, and there's no clear boundary between the colony and the edge of the colony. This works for about 95% of bacteria and 95% of fungi. So if you notice this, that's a fungal colony, there's a fungal colony, and these are all bacterial colonies. Any question about that? You'll note the bacteria have different characteristics, meaning the different colonies have different characteristics. There can be a different shape to the colony. There can be circular, rhizoid, irregular, filamentous, and spindle. The margin is the edge of the colony. There's an entire margin where the edge is smooth. There's undulate, lobate, and curled. And I'll never ask you, is this margin undulate, lobate, or curled? But I will expect you to be able to distinguish between an entire margin and one that's either undulate, lobate, or curled, as well as I can ask you the difference between these three and these two. But I would never ask you, is this colony margin rhizoid or filamentous? Because those are too similar to each other. Any question about any of that? Elevation is the elevation of the colony. And to see the elevation, you literally have to take the lid off and then look at the colony from its side. You're going to have a flat colony, a raised colony, convex colony, a pulvinate colony, that's a superconvex, and then an umbunate colony. I will never ask you to distinguish between a convex and a pulvinate colony. In fact, I don't think I even ask you ever to distinguish between the elevation of the colony because it's too difficult in an online class to get the elevation. And then there's the size of the colony. Punctiform is a pinprick of a colony. That's the smallest colony you can see. A small colony is about the size of a pencil or a pen, tip of the pen. A moderate colony is one between the tip of the pen and the eraser side of the pencil or pen. And then a large colony is one larger than the eraser of a pencil. Any question about any of that? Texture is whether the colony is smooth or rough. Surprisingly, students have a little bit of trouble distinguishing between a smooth and rough colony. The appearance is whether the colony is shiny or dull. Usually students don't have too much trouble with that one. Non-pigmented is if the colony has no color. That would include a tan colony, a white colony, an off-white colony, a cream-colored colony, versus a pigmented colony, one that has color. Purple, red, or yellow are frequently pigmented colonies. And then a colony can have an optical property. Most colonies are opaque. A few are translucent. Very few are transparent. Transparent means you can see right through the colony like you can see through clear water. Very few are transparent. Let me show you an opaque colony. This colony there, right by the B there, is opaque. See, translucent colony. There's a translucent colony, one that you can kind of see through, but it's not crystal clear. You can see through it like seeing through slightly muddy water. Generally, when I ask you the questions about the colony, I expect that you'll know something that's simple, circular versus irregular. um, punctiform versus small, smooth versus rough, shiny versus dull, pigmented versus non-pigmented. The only one that you probably should learn is punctiform because that's not a term you might be familiar with. And what other one? But all right, there are two terms you must understand for this lab. Abundance, that is simply the total number of colonies on an agar plate. It's got too many there, so I'll go to one that has fewer. So if we were to look here, we could count all the colonies, although it'd be really difficult counting them here, here, and here. So we'll just say, what's the abundance of the colonies in this region of the plate? I'm going to call 12 there, unless that's a colony there, in which case it's 13. Okay? And that's the abundance. The diversity is the different colony types. Now, in this region right here, what's the diversity? 1, 2, 3, 4. What's the diversity? This region right here.
2.
Yeah, 2. This colony type and that colony type. So the diversity is the different colony types. And you can determine the different colony types by looking at the differences in the colony characteristics. Like right here, I can see there's clearly in this region right here, 3 different uh, colony diversities, that one, and that one. Now, sometimes it won't be clear, like this here and this there. Is that the same colony? Is that one there and that one there? It's not entirely clear. So sometimes there's a little bit of wiggle room on what the diversity is. And generally, as long as your number, when you say the diversity, as long as your number is close, I will allow it. But if you say 4 here, I'm going to say that's not close enough. Okay. And obviously, if you only say 1 here, I'll say that's not close enough too, because that's clearly 2 there. But on this plate here, we got 1, 2, 3, 4, 5, 6. At least 6 different types, maybe 7 if these are different. Okay. So the diversity on this plate is at least 6, maybe 7, maybe even 8. If you count something like something in here. But if you were to go like 15, I'll say it's not close enough. And if you go 4, I would say 1, 2, 3, 4. Just right here alone, there's 4. And that's obviously 5 right there. And that would be probably 6. Any question about that? So when I ask for colony abundance and colony diversity, you don't have to be exact, but you should be close. I have another example down here. This one's very easy. In this image, what is the abundance of wild cats? Come on.
7.
Yep. And what's the diversity of cat species?
2.
2. Lions and cheetahs. 5 cheetahs, 2 lions. All right. Any questions about any of that? Know the terms of this chapter? You do not need to go through, that's kind of an interesting picture. You do not need to go through the references. And let's go to the questions of lab one. Remember, if you're submitting a lab, to always submit the worksheet, not the lab module. The first time I'll give you a warning. After that, you'll get a penalty. Any question about that? The reason being, if everyone were to submit a lab module, my web page would soon become full, and then nobody could submit the lab module. All right. In this lab, everything's going to be done online. Actually, there might be some reading I need to talk about. Or did I give that already? I must have given that already. Ah, this one, there's no questions in the lab module. I removed them. All right. Protocol one. We're going to look at a number of different environments for inoculating general purpose culture media with the following: one, air; two, your lab bench; three, your hands; four, your lips and your tongue; and then five, from the soil. So in procedure one, what is in the air you breathe? Below is a nutrient agar plate that was exposed to the air and incubated for 24 to 48 hours at 30 degrees Celsius. And what's the colony abundance on this plate? Answer that here. What's the colony diversity on the plate? Answer that there. And then C, 1C, what step would you take to reduce airborne contamination of a sterile agar plate while you are inoculating it? So while you're inoculating a plate here, and you remove the lid, what steps can you take to keep the plate from getting airborne contamination, something falling down from the air? So don't tell me something that you can do after you inoculate it or before you inoculate it, something you can do while you are actively inoculating it. Procedure two. What is on the arms when you put your arms on your lab bench? What we do is we inoculate the lab bench and then disinfect it. So here is a plate showing you the growth of a plate, which isn't showing up very well. Let me blow that up. Before we disinfect the lab bench, and then here's a plate after we disinfect the lab bench. First question for procedure 2A, did table disinfection work? You just need to answer yes or no. And then 2B, what is the purpose of allowing the table disinfectant to air dry? Or why do we air dry the disinfectant? Answer that there. Procedure 3, what is on your hands? What we've done is we touched a plate with hands before we washed your hands, right and the left. And then we touch the plate with our hands after we wash your hands and you dry your hands. The first question is, does hand washing work? You can answer that yes or no, or you should discuss it a little bit, because it didn't work 100%. And you do need to dry your hands because the water from your hands will wick the bacteria from your hands onto the plate. And when I was first teaching microbiology in the lab and actually had my students doing this, some students were getting more microbes on their clean hands, the hands they wash, than the hands before they wash them. And then I asked them, did you dry your hands? And always, they never dried their hands. So the water on your hands can wick the bacteria from your hands to the plate. So you need to dry your hands. And then two, suggest one reason why hand washing did not result in the complete removal of microbes. Hint, it may have to do with the normal microbiota on your skin. If you obtain an answer from research, please provide a citation. This is not simply the state. You can't simply state, we have normal microbiota on our hand. You need to state why the washing did not kill all the normal microbiota. All right, procedure four. When you kiss your significant other, what exactly are you giving him or her? What's on your lips and on your tongue? So we're talking about French kissing here, huh? All right. Here is the plate that somebody kissed the plate with their lips, and here's a plate where somebody pressed their tongue on the plate. Please note that I have a note up here above the picture. You should read that before answering uh, the questions for procedure four, sorry. Which plate, procedure 4A, which plate has higher colony abundance, your lips or your tongue? And then B, why do you think this is the case, even though you might expect the abundance of both body sites to be the same? I mean, they're physically close to each other, and you probably often lick your lips with your tongue. So what might cause the observed difference in the colony abundance? I'm going to warn you that I will require two answers here. There are three answers that are very easy to get. I've actually had students suggest a total of something like five answers, but there's three that are very easy. And you must have two answers to get this one full credit. Any question about any of that? And then procedure three, why was the tongue plate incubated at a higher temperature than the lip plate? The lip plate was incubated at 30 degrees, and then the tongue plate was incubated at 37 degrees. Procedure five, are microorganisms in the soil? Here we have a plate where we took some soil, put it into a tube of water to dilute the soil, and then smeared the water on the plate. And what is the colony abundance for this soil plate? And you can give an answer there. And then the second question, is it even possible to accurately answer this question? Please explain your answer. And then 5B, is there greater abundance of colonies from the soil compared to the lip plates? So there's the lip plates. You'll note that there are some big colonies on the lips, most are small. And it may look like all of the colonies are large here, but if you look closely, you can find a number of small colonies as well. Like there's a large colony, there's a large colony, but here's a small colony, there's a small colony. And there's some very small colonies right there. And it's impossible to say how many small colonies are right here. So there are some small colonies on the soil plate. All right, any questions about this lab? If not, I will be here until 8 o'clock to help you with the lab. Go ahead and begin working on the lab. If you have any questions, please ask. Once the last student is logged out, I will log out as well. So if you have a question, make sure you log in early in the lab. All right, any questions?
Yes.
Yeah,
I do have question about the extra credit for the
okay.
one that you want us to do the schedule
Uh-huh.
thing. When I download it, it's just so only the input my schedule, not the
You mean you
study.
only have one page?
Yeah, just only one page.
You're not using Excel, I take it?
I download what you posted.
Do you have Excel on your computer?
Yeah,
Then you should get another tab.
sure.
If you do not have Excel on your computer and you're using another worksheet processor, you may only get one page, in which case you'll need to make another page. Just copy it and make another page.
I see. Thank you.
All right.